have a timer, but then you have a timer there too. So, and then 1 minute prior to finishing, so before you wrap it up, and then we're going to have a Q&A, and you have fun.
Oh, you sorry about that.
Good afternoon, everyone. We are team INSA-UPS from France, and we are proud to present our project, Croc'n Cholera. All the synthetic biology projects want to take advantage of the microbial diversity and the incredible capacities of microorganisms. Most of them want to do so by engineering one single organism. But we want to do things differently. We think that each organism is specialized in its function and is the best at it. So we want to take advantage of them separately. We want to combine them for a better efficiency towards a single purpose. For that, we want to engineer a microbial consortium. We want to engineer each organism to adapt or modify its function according to its capacities. We want to then engineer them further to be able to combine them. For that, the first step is to process information between different kinds of organisms. That can be two prokaryotes, or a prokaryote and a eukaryote. But we want to process information to achieve a specific functionality. That can be a stimulus processing, a transmission signal, or a response, making each organism either a detector, a relay, or an effector. But to prove the interest of our concept, we wanted to apply it to a concrete problem. For that, we set our focus on the cholera disease. As you can see on this map, cholera is still a huge problem worldwide, affecting many countries. And we can say that the prevention systems currently available are not efficient enough, because there are still up to 4 million cases per year according to the WHO. Cholera is caused by the pathogen Vibrio cholerae in contaminated water. That's why we applied our concept to Vibrio cholerae. We want to be able to detect it and kill it in contaminated water. For that, we decided to use CAI1, its specific quorum sensing molecule, to detect it. According to our concept, we chose the best organism to detect it, and we chose a non-pathogenic Vibrio, Vibrio RVI, that already has all the quorum sensing mechanisms and could be able to detect CAI1 instead of its own quorum sensing molecule with a minimum engineering. The other essential step of our project is to be able to kill Vibrio cholerae. For that, we decided to produce AMPs, antimicrobial peptides, able to kill bacteria. For that, we decided to do something that has never been done before, which is express AMPs from the crocodile immune system that show a specific activity against Vibrio cholerae. Again, according to our concept, we chose the best organism to produce those AMPs, and we chose Pichia pasteuris, which is a yeast, a non-robust producer of proteins that won't be affected by the AMP activity. As you can see, we need to complete our system. We need to transmit the information between Vibrio RVI and Pichia pasteuris. For that, we used dicetyl that has already been used by a previous iGEM team. So we want to engineer Vibrio RVI to produce dicetyl upon detection of CAI1 and Pichia pasteuris to produce AMPs upon detection of dicetyl. With that, we designed a synthetic microbial consortium against cholera. But this is a complex consortium with many biological entities interacting together. So we needed modeling to achieve a rational design and to check the feasibility of our system before going headfirst into wet lab. Firstly, we decided to represent our model with an SBGN notation to facilitate the discussion between all the stakeholders of our project. You can find here all the parts previously explained, so Vibrio cholerae in red, Vibrio RVI in blue, and Pichia pasteuris in green. Then we decided to solve our model to assess different parameters of our system, and we obtained those graphs. So first, we checked the feasibility of our project. And the model showed a rapid decrease of Vibrio cholerae under the pathogenic level, and as predicted, Pichia pasteuris is not affected by the activity of AMPs. The model predicted a response time of about 1 hour, which means that the water could be drinkable in about 1 hour. Then, we checked the real-life usability of our system. As you might guess, there might be fluctuations during manufacturing process or during transportation. So we checked different sets of parameters around the reference values, and we obtained a little deviations of 3 minutes, which shows a robust behavior of our system. Finally, we decided to have a deeper understanding of our system to be able to optimize it. And we found that the response module is the one that could be optimized. The model was proved successful and our design was validated. But this is an iterative modeling that needs data from the wet lab.
Okay, so now I'll present the results we obtained during our 4 months of experiments. So first of all, because of safety regulation, we couldn't use Vibrio cholerae in the lab. So our first module focuses on mimicking Vibrio cholerae thanks to E. coli through the production of the gram-sensing molecule CAI1. But due to some uncertainty about the use of Vibrio cholerae genes, we decided to use the gene of Vibrio arvei, a non-pathogenic Vibrio strain. So instead of producing CAI1, E. coli will produce CAI1 from Vibrio arvei. This gram-sensing molecule is synthesized by the enzyme CQSA, so we therefore engineered E. coli to express CQSA. So once the transformation done, we had to detect CAI1 production from E. coli. We were expecting 2 specific shifts in NMR, but the analysis couldn't provide them, either in the assay, neither in the positive control. It was therefore not possible to conclude anything about this part, but probably because CAI1 have been produced below the sensibility limits of NMR. Because it's known that gram-sensing molecules are often produced and active at low concentration. So we decided, it's not working. So we decided to do an in vivo detection because CAI1 induces bioluminescent genes of Vibrio arvei. So we used a Vibrio strain deleted from the CQSA gene, so it cannot produce its own gram-sensing molecule, to know whether the supernatant of E. coli could restore the bioluminescence. So you can see that the supernatant coming from the wild-type Vibrio arvei, which is the positive control, does restore the bioluminescence, as well as the supernatant coming from the engineered E. coli, while wild-type E. coli does not induce any bioluminescence. So CAI1 has been efficiently produced by E. coli, and this way we've been able to establish for the first time synthetic communication between E. coli and Vibrio arvei. Next, CAI1 will trigger Vibrio arvei to produce diacetyl. But first, we needed to learn how to modify Vibrio arvei since transformation does not work with it. Indeed, the only way to bring in modification is through triparental conjugation. So we did preliminary tests using the RFP gene, and as you can see, we ended up achieving genetic modification of Vibrio arvei. To the best of our knowledge, the first time Vibrio arvei is used as a genetic chassis in iGEM, but also the first time RFP is proved to be functional in this bacteria. So once we managed the triparental conjugation, we had to engineer Vibrio arvei to detect C8CAI1, but also CAI1 from Vibrio cholerae. And for this, we took advantage of a mutated CQSS receptor able to detect both molecules. So the next part is to trigger diacetyl production when CQSS is activated. So the second genic part is composed of PQRR4, which activation leads to TETR production and PTET inhibition. Once CAI1 activates CQSS, PQRR4 is inhibited, leading to the activation of PTET and to the production of ALS. And ALS is the enzyme which enables diacetyl production. These 2 parts have been successfully cloned in E. coli, but they still remain to be conjugated in Vibrio arvei. So we tested in E. coli if ALS expression indeed allows diacetyl production. So using an NMR experiment, we observed a specific shift at 2.34 ppm when the supernatant was artificially supplemented in diacetyl. That's the red curve. There is no signal in the negative control in green, while in both duplicates of E. coli expressing ALS, in blue and violet, you can observe a weak peak coming from the diacetyl. So this part is validated, but still remain to be conjugated and tested in Vibrio arvei. Next, Bishop Asterisk will detect diacetyl thanks to the ODR10 receptor coming from C. elegans. So once activated by diacetyl, this receptor will process the signal into antimicrobial peptide production through the first one promoter. Every part has been successfully integrated in the yeast genome. So using the RFP gene instead of the AMP gene, we check the activity of the ODR10 p1 pathway. You can see that the increase of diacetyl put in the culture media is correlated with an increase of the RFP fluorescence, showing that ODR10 can detect diacetyl, but also that we have been able to activate its downstream signalization cascade, leading to antimicrobial peptide production. But because these AMP have never been produced before, we wanted to assay the mRNA expression of this AMP. Using an RT-qPCR experiment, it will show that DNY15, one of the 3 AMP tested in our project, is well expressed, while there is no specific amplification of negative control with water or the antiplasmid. So once we have this confirmation, we aim towards cytotoxicity tests. To do so, discs were soaked in Bishop Asterisk supernatant and then put on solid culture Vibrio RVI. And it will show that DNY15, again, inhibits Vibrio RVI growth. You can observe the inhibition halo around the disc where there is no bacteria growing. So the condition to prove the functionality of the two other AMP have not been found yet, but we still managed to efficiently produce DNY15. This is the first in vivo production of this peptide, and also the first successful AMP production of this peptide by iGEM team. So DNY15 is also the first crocodile gene submitted to the iGEM registry. Now, to sum up everything, we performed a lot of experiments this summer, and we are proud that most of them succeed. We established for the first time synthetic communication between E. coli and Vibrio RVI. We managed to do the triparental conjugation in Vibrio RVI, but our parts still remain to be integrated. We managed the activation of the ODR10 p1 pathway in Bishop Asterisk for the first time. And last but not least, we demonstrated that an engineered yeast can produce efficient antimicrobial peptide coming from the crocodile. So integration in Vibrio cholerae in Vibrio RVI will take a bit more time, but as you can see, we are close to end our project. And using the cholera topic as a proof of concept, we will reveal how useful synthetic microbial communities can be to tackle new issues.
So, as we were dealing with the cholera disease, it was clear for all of us that we didn't want our project to stay in the lab. That's why we decided to put a huge effort on its application. So first, we interviewed members of NGOs, non-governmental organizations, to have more information about the cholera situation. We learned that in developing countries, people can get ill while drinking from contaminated water source. But NGOs are already on the field and can immediately provide curing treatments and water treatment products whenever it's needed, but each of them have their own issues. Indeed, bleaching, boiling, and filtering are sometimes not adapted to local population customs, and more importantly, they cannot be easily used in remote villages. That's why NGOs are always looking for alternative solution, but only if the usual product life cycle pathway is respected for these products. Indeed, the following steps have to be considered: production, providing, distribution, daily use, and waste treatment. So our team decided to analyze each of these key steps thanks to a pragmatic tool called the ethical matrix, especially suitable for new technologies. So we defined 8 stakeholders and 3 human values important to us: well-being, autonomy, and fairness. Eventually, we got 3 main outcomes from our analysis. So first, our idea must be as user-friendly and trustworthy as possible. Second, it is important to educate members of NGOs and also daily users to our non-conventional technology, for example, through a user manual. And third, it is important to avoid any GMO leakage at all the steps of a regular product life cycle. Now, the next step for us was to find an industrial partner to help us build a physical device based on our idea. So indeed, we made a partnership with a startup in Toulouse called Sun Water Life and specialized in water treatment products. We encouraged them to develop a research project for their firm based on our own, and they helped us financially and technically. As a result, we came up with this. So we thought to freeze dry our microorganism into a little bag made of sun-water-like membrane, and then put it into a tube-shaped container made of ABS plastic to let the device float on water, and enclosed it both ends by the same membrane to allow molecular diffusion. Once in water, current-sensing molecules from cholera would be able to come in, and antimicrobial peptides would be able to come out. The water treatment process over, it would be possible to open the container, pick out the tea bag, burn it, and put another one in the device to treat another volume of water. We were actually able to evaluate the performance of our prototype that we printed in 3D thanks to the model we described before. So from a little bag of 10 centiliter containing a freeze-dried biomass of 7 grams, it will be possible to treat 5 liters of water in just 1 hour. So based on this prediction, we carried out a business analysis thanks to the deputy director of the business incubator in our area. We realized that our company should have a specific economic model to gather and integrate all of the stakeholders gravitating around our project. Also, the best option for us would be to provide a service. That is, the ability to treat 57.5 cubic meter of water, and also training sessions for 5 members of NGOs. Now, while developing our ethical and business analysis, we soon realized that people lack huge scientific knowledge key to our project: microbial diversity, life engineering, and synthetic communication. That's why we decided to empower people with science, working on 3 different milestones. The first one, discover. The second one, practice. And the third one, discuss. We carefully thought about our educational approach and developed a precise methodology based on bibliographical research. We then applied them on the following examples. We made people discover microbial diversity during primary school interventions and also during exhibitions we organized in our academic libraries, not to mention press articles written about us. We made people practice biology during scientific events held in our town and in our city, and also during a high school lab work we entirely supervised. We regularly talk with people about our project, especially during a seminar we organized, thus efficiently raising interrogation toward our project and toward synthetic biology. But we are also especially proud of the card game we designed this summer called Microbi World, entirely dedicated to make people discover microbiology and microbial interaction in a playful way. The 55 card of our projects all of them are inspired by scientifically accurate biological features. And I got a big news for you. If you iGEMers are interested in our funny and witty games, you can come and see us at our exhibition table, because it's available here on Jamboree, table 28. Now, I think you saw in this presentation that we made a lot of work during our project, and maybe Medal Croc'n would be a good way to sum things up. So we think that we're suitable for the gold medal and for 5 different prizes because we checked all of the requirements. We are proud of our wet lab work because we managed to characterize 5 existing biobricks and also obtain 6 new ones, validating half of them while working on 3 different microorganisms. But we are also proud of our dry lab work, modeling, entrepreneurship, or human practices. All of them combined together convinced our industrial partners, Sun Water Life, to continue the project after the end of the competition. So we would like to thank all of the team that collaborated with us this summer. Some of them are here, I can see you. We would like also to thank all the people that helped us, and also we would like to thank all of the sponsors who trusted us with our crocodile project. Thank you very much.